Hi, my name is Nenad Bursac. I'm a professor of biomedical engineering at Duke University, and I was moderated session today at the American Heart Association BCDS meeting on cardiac regeneration. I was really intrigued by a talk of Dr. Minxia Gu from Cincinnati Children's Hospital, who told us her really interesting work in iPS cells and derivation of endothelial cells for studies of congenital heart disease. So, Dr. Gu, what's your summary for your work that you've shown us today?
Right, so in our research group, we're really interested in generate these human iPS-derived organoid and endothelial cell models to study congenital heart disease and congenital lung disorders as well. And our motivation is mostly because there's really no good animal model to study congenital heart disease. And we don't really understand what's the genetic variance that is, causal to these conditions. So we took patient iPS-derived endothelial cells to study the endothelial and endocardial abnormality in the hypoplastic left heart syndrome. And using these human patient-specific iPS derivatives, we're able to uncover new disease mechanisms. We found there's abnormal endocardial gene expression that leads to abnormal valvular development, coronary functions, etc. in this condition.
Yeah, I found it very interesting. So I specifically you showed that fibronectin is very important and potentially downregulating in hypoplastic left heart syndrome.
Right.
So is that something that we could in the future target therapeutically?
Yeah, that's definitely a very good point. So we did a little bit of exploration in vitro using the stem cell models. What we did was we co-cultured human iPS or patient-specific iPS-derived endocardial cells with myocardial cells. And we found if we co-culture even normal cardiomyocyte with patient endocardium, these myocardial cells became abnormal in terms of cell proliferation, contractility, etc. And we found when we added fibronectin into the culture medium or into this co-culture system, it actually rescued the cardiomyocyte functional defects. So solely from the in vitro perspective, we think it's very promising to move to the next step.
Yeah, and I was really intrigued also with your lung-specific endothelial cells. That's been the first time that this has been derived, the protocol for derivation. So where do you see is a future use for these cells?
Yeah, we're actually very, very passionate about this project because for us like working in the endothelial cell field, we know one technical challenge is that when we take endothelial cells out from their in vivo environment, they lose their organ-specific features even within one passage. And we found when we co-culture or co-differentiate endothelial cells within this organ-specific microenvironment, and in our cases provided by organoids, they could maintain and gain organ-specific features again in a culture dish. And going forward, we think one technical challenge is to perfuse these vessels, because perfusion is really important for maintaining normal endothelial cell function, because otherwise the vessel organoid could regress after a couple of days if there's no perfusion. So we're currently working on connecting the vascularized organoids with a microfluidic device to perfuse these vessels to let them further mature.
So kind of organ-on-chip systems.
It's kind of organ-on-chip. We also did in vivo transplantation. We transplant those vascularized lung organoids in a kidney capsule. So there's circulating blood, perfused the organoids, and we found they further matured for another 3 months in vivo. And the interesting thing about this is that the vessels that we created within the organ system, they maintain their lung-specific and human-specific features. And the mouse vasculature only exists at the border of the organoids, which is quite different from traditional approach. When they transplant organoids, the mouse vasculature invade into the human organoid. So in our case, it's really human vessels.
Great.
Yeah.
It's an example of really innovative work, so congratulations.
Oh, thanks so much.